Okay, here we go. So let's go ahead and get through this. So cool, don't care. Um, don't really care. This is a cool slide. It looks terrible. But I'm going to be honest with you and say it's not that bad. There's a lot of stuff that goes on in here, but here's all the names. If you are interested in what you're learning, so you guys will probably be covering this, a lot of the TCA stuff, carbs, not really, not really, not really. So that's pretty much it. This, and then this. In 441, you guys will do purine, pyrimidine, amino acid stuff. So I would focus on these for now. The MCAT covers, if you're interested in the MCAT, it covers a decent amount of these, but I wouldn't be too panicked about these. What you'll notice about these actually is that a lot of them interconnect. So you'll find that the products for one end up going to the other, or like something from Krebs cycle actually suddenly can turn into an amino acid and stuff like that. So the way that you make connections is by trying to see how the structures are similar, and the more similar the structures are, the easier it is for you to connect them, right? So if we go down here, don't care. Don't really care. Again, more PV to glycolysis, but I don't really care that much. What I want to take you down to is this slide, and like the slides following it. But basically, spontaneity is something that comes up a lot in 440. I want you to recognize that a lot of the reactions that you do in the 44X series are not spontaneous. So steps 1, 3, and 10 in glycolysis are irreversible. Especially step 1, for example, as shown here, is endergonic, so it requires energy. And the way that we do that, get through that, is by coupling it to ATP. So like you can see here, if you look at this, right, we go from final, which is a positive number up here, to a lower number down here. So if you remember, we do the G final, right? So we do final minus initial, right, is going to be equal to whatever our energy is for that reaction, right? If it's negative, it's spontaneous. If it's positive, it's non-spontaneous, right? So you should be able to tell me from this reaction, because we're increasing in delta G, it's a positive delta G. This reaction normally is endergonic and requires energy, right? That makes sense for everybody? Yeah? Okay, cool. Now, how do we get by that? We get by that, yes? For now. So by like let's say they didn't give us the diagram. Yeah, okay. Um Probably not. That seems a bit mean. Yeah. Calculations though they will make you do. So they will give you I don't know if you've gotten the new equation, the delta G naught plus RT ln Q yet. Okay, so don't worry about that. But that equation, you will be required to do calculations through that without a calculator, right? So please, I beg of you, please get used to doing mental math, or at least doing simple like calculations. So like log of 10, log of 100, log of 0.1, log of 0.01. Log of 3, right? Being able to do those logs makes life a lot easier, and it makes the class so much easier, significantly easier. There's only 2 things that make the class easy, in my opinion. 1, memorizing the amino acids really early, and 2, being able to do math like that. Right? Makes the class 10 times easier. But yeah, anyways, for this, right, it's an endergonic reaction, right, because we're going from a low delta G to a higher delta G. A positive delta G is non-spontaneous in this case, right? So if it's non-spontaneous, that means it doesn't happen in our cells, right? Wrong. It does happen in our cells. And the reason it happens in our cells is because we couple it to ATP, right? Coupling is very important, right? Because if we don't couple things, we just die. So what you'll see here is the hydrolysis of ATP produces this massive energy drop, right? And if we add this reaction to the previous reaction, we would ultimately get a reaction that is very negative in delta G, right? That is what allows this reaction to actually occur, and that is why this reaction is irreversible when you actually look at it in a cell. That makes sense for everybody so far? Cool, okay. And here's just a slide kind of detailing that again, right? That's all they're showing in this slide, that if you add these 2 together, you get this, right? And that's how this whole process is spontaneous. Okay. Um, this slide is really weird to look at. I don't know how they explained it in lecture, but I think the easiest way to think about it is that in any given reaction scheme, you'll have your substrate, you'll have some high energy transition state, you'll have an activation energy, and then you'll also have obviously your product. And then when you add the enzyme, what tends to happen is the enzyme substrate complex will form, which is quite stable. The activation barrier between this activated state and the ground state is now lower. The ground state is technically here, actually, right? Is now lower compared to this reaction scheme over here, and you get a product. All the enzyme does is just make the reaction easier to do, right? This is different from kinetics, though. Kinetics is different from equilibrium, right? Delta G is not the same as the rate of a reaction, right? The K of a reaction. I don't care about any of this, right? We'll cover proteins in depth when you guys actually start talking about proteins. But if you want the in-depth stuff for this, the videos will cover every single slide, right? So, yeah, moving on. Yeah, so this is just an overview of what you guys are doing. I would say the most important one out of these is this. This is the enzyme kinetics and mechanisms is where most of the math will happen. And it can get very confusing very quickly if you don't understand the concepts and what is going on. So I would pay special attention to that and just keep that in mind. The pathways, you just have to memorize them. There's no ins and outs. Get your Anki out. Memorize the pathways. The pathways have to be memorized. You'll realize that some things are very connected. So like glycolysis and glycogen metabolism are actually quite connected. Citric acid cycle as well is like once you understand the reaction mechanisms, and this is why I say OChem is really important. If you understand the reaction mechanisms, everything else becomes way easier. Same thing with oxidative phosphorylation and the pentose phosphate pathway. The pentose phosphate pathway is a bit different. There's some weird stuff that happens there. But generally speaking, if you can understand the mechanism, the underlying mechanism for every reaction, it becomes way easier and you will do better on the exams. 441, you guys cover a lot of lipid and nucleic acid breakdown and stuff. I actually really liked this class. The professor sucked. But I really like this class. You guys cover fatty acid oxidation buildup, lipids, nucleotides, amino acids. I really like this course. But yeah, and 442 is 442 requires a lot of experimental design. Especially if you take it with like Rahola Baker, for example, she absolutely loves forcing you to do experiments. Like she'll give you a list of 20 different experimental techniques and she'll ask you for different types of experiments, what order would you put these numbers in to actually get a result for this experiment? That was the first page of my final exam. So yeah, there's that. But don't worry about that right now. We'll focus on the pathways first, and when we get to 442, we'll get to 442. But that is all for that first lecture. Now the real content starts. Where is 2? Okay. This is where I feel like people start getting lost and terrified. So let's try and make this a bit simple. Again, I'll just try and go over stuff that actually matters. Okay. pH. pH is very important. Why is pH important? Aside from the amino acids, which I don't expect you to have memorized, but I suggest that you memorize them as soon as possible, right? Molecularly, when we change the pH of our system, right, the amino acids also get altered. When we alter the amino acids, we alter the proteins, and the proteins fold in different ways. And if you misfold or change the folding of the protein, it will most likely break, and that'll break down other biological functions, right? So we talk about like medical conditions like acidosis and alkalosis, right? Basic and acidic conditions, right? Under acidic conditions, you break your proteins. Under basic conditions, you break your proteins. Usually leads to death if you don't quickly fix that. But your body is really good at regulating that, and thank God it is, because of the bicarb buffer system, which we'll talk about. This is just saying that proteins have different shapes under different pHs. That's all there is to it. There's nothing much to that. But yeah. Where is the next slide? Yeah, okay, so just a brief review of acids and bases, right? I think most of us have taken OChem relatively recently, or GenChem relatively recently. But the general idea is, right, that a strong acid like HCl or a weak acid like acetic acid, right, will dissociate into a proton and its conjugate base, right? That's all there is to it. And when we make the expression for that, right, which you will have to do, right, when you're doing delta G equations, you may have to do expressions like this. It won't be Ka, though it'll be Keq, right? But we do products over reactants for that, right? And then we plug that into some expression or whatever, and then we solve for delta G if that's what we're looking for. But that makes sense for everybody so far, right? Dissociation of acids are okay, right? Same thing for a base, right? It doesn't change for a base, right? It would just change into, instead of HCl, it'd be, I don't know, like NaOH, right? And it would be sodium and then hydroxide, right? Good? Okay. What to remember about acid dissociation constants. So when you start memorizing the amino acids will have different pKa's. What's important to memorize about these is that if you are below the pKa, you're in a protonated state, right? It does not always mean it's positively charged, right? Like, for example, a carboxylate ion, right, is negative when it's in base and neutral when it's not in base, right, when it's an acid, right? An amine is positive under acidic conditions, but neutral under basic conditions, right? So keep in mind that just because it's acidic does not mean it's positive, and just because it's basic does not mean it's minus, right? The other thing you should know is that a higher Ka is a stronger acid, right? So it dissociates more, right? That makes sense, right? Because if we have more of these two things, right, that's the dissociated form of any given acid, right? That makes sense. The pKa is the inverse, and I suggest you memorize these equations if you don't have them memorized. They will be helpful just because I don't remember if you get an equation sheet for biochem, but you only have 50 minutes for an exam, and being able to do that math quick is helpful. And you have a question, yeah?
But we still consider like basic amino acids as having a negative charge on the side chains, right?
No, that's the thing, right? So lysine and arginine, right? Yeah, positive, right? So lysine and arginine, right, are basic. They are not negatively charged under physiological pH, right? Under physiological pH 7, they are protonated. They are positively charged, right? So lysine's pKa is about 9.6, and the arginine's is about 12.1. The pH is lower than the pKa, right? pH is 7, pKa is 12.1, 9.6, right? Therefore, it is in the protonated state, and because it is an amine, protonated state is positively charged, right? You can do the opposite for aspartate and glutamate, which are carboxylate groups, right? The pKa is for those are about 3.6 and 4.2, right? If you go above that to, let's say, 6, right? Normally, in the acidic form, they're neutrally charged. There's no charge, right? They're neutral. But as you go above that pKa, you remove the H and it becomes negatively charged, right? So the things that are acidic have a negative charge, and the things that are basic have a positive charge, which is completely opposite of how we were taught them in chemistry, right? But that is because we're always thinking about it from the perspective of being in physiological pH, right? That makes sense for everybody, how we're doing this? Yeah? Okay. But yeah, I would just say for this, try and keep these relationships in mind because they could very easily ask you questions about this. The Henderson-Hasselbalch equation, I honestly just memorize it. Just know this equation. Don't bore yourself with trying to derive it. I mean, I don't think they've ever asked me to derive an equation like this. So, yeah. Okay. Buffers. I feel like this is probably the thing that gives people the most pain. Buffers are weird, but I'll try and make it a little bit simpler. So for these curves, right, there's two main things that you should keep in mind, and I don't really have, is there a better example of this? Not quite. But the one thing you should keep in mind for these is where do I find the midpoint and where do I find the equivalence point, right? And what the heck is an equivalent, right? So the first thing we should talk about is what is the midpoint? Easy enough. The midpoint is the pKa. So if you have an amino acid, for example, right, whatever the pKa of that amino acid is, will be the midpoint of the titration curve, right? And the midpoint is this, right? So for this example here, the midpoint is 4.76, right? And there's a couple relationships that you should keep in mind, and I can't draw on this because I suck, but if we look at Henderson-Hasselbalch, right, I want to make something clear, and it's just this idea that by plugging numbers into this expression, you can find yourself anywhere along this curve, right? And the way that we know that the pH equals pKa is the midpoint is that the only way that's going to be true is if the concentration of the base is equal to the concentration of the acid. There is no other option for that, right? So for example, if the amount of base is 1 mole, right, and the amount of acid is 1 mole, 1 divided by 1 is 1, and the log of 1 is 0. And by that logic, pH has to be equal to pKa, right? So what that should tell you is that in order for the midpoint to be true, there must be equivalent amounts of the acid and the base. That makes sense, right? Cool. As soon as you deviate either side of that, right, now you're entering either basic or acidic conditions, right? So for example, if I up the base, right, I will be on this side of the curve, right? If I up the acid, I would be on this side of the curve, right? Good? Okay. The other thing to keep in mind is what the heck is an equivalent? Simply put, the equivalent is just a universal way of measuring things. So, for example, if I have 1 mole of base, right, and 1 mole of acid, an equivalent would be 2 moles, right? 2 moles of OH minus, or 2 moles of H plus, or whatever, right? An equivalent is just the total amount of moles of said acid slash base in a solution, right? And that makes sense because if we look at this graph here, right? Let's say that we have 1 mole of substance, right? Just 1 mole of 1 mole of HA, right? So 1 mole of some acid, right? If I add half of an equivalent of this, I would be adding 0.5 moles of hydroxide, right? When I add the hydroxide into my solution of purely acid of 1 mole, I will turn 50% of that 1 mole of acid into 0.5 moles of base, right? Because that hydroxide is ripping off a proton from half of those acids, right? Does that sort of make sense? This is a really weird concept. It's not easy to grasp. So if you don't understand, please tell me and I will try and explain it again. Yeah, you want to explain it again? Okay. So if, for example, I have a solution, maybe if I draw out like a nice little picture here. If I have a beaker, right? And let's just say I have 2 acids, right? I have 2 acids in here, right? I can't draw, right? So I have 2 molecules of acid in here, right? If I add that's my equivalent. So two amounts of acid is my equivalent. Let's just say these are moles, right? So 1 mole of acid here, 1 mole of acid here, right? So I have 2 moles of acid, right? Cool? That's good so far, right? If I add 2 moles of base, right? So 2 moles of hydroxide, you would agree that I would remove both of these protons, right? And I would just have a beaker that just has A minus in it, right? There's no HA in there, right? That makes sense? Yes? Okay. The idea of an equivalent is just how much stuff do I have to add to turn the solution from completely acidic to completely basic, right? So this number changes. So for example, if I have 3 HAs in here, right, I will require 3 moles of OH minus. Or if I have 4 Oh wait, 4 acids in there. I will require 4 OH minuses. It doesn't matter though how many I add. That is one equivalent for that specific solution, right? What I'm trying to say is that it's a moving target, right? It's not constant. It's just not a constant, right? It changes depending on the solution that you're in, right? Yeah? Does that make sense? It makes more sense now. Okay. Yeah.
What about for like polyprotic acid?
Right. So that's a big question, right? What about for polyprotic acids? The whole point of using equivalents, and you'll see this when you do amino acids, is amino acids have 3 pKas. They don't have 1, they have 3, right? Things like arginine, aspartate, glutamate, they have 3 pKas, right? So when we go from one form of the amino acid to another, so like let's go from, let's say, if you guys remember stuff from like OChem, that's the thing that matters, but let's go ahead and look at this example. So what's an easy amino acid here to keep in mind? Let's go with like, I don't know, aspartate, right? So aspartate looks something like this. This thing has 3 pKas on it. There's a pKa for this group, a pKa for this group, and a pKa for this group, right? So you basically take this graph and multiply it by 3 times, right? Because there'd be 3 different pKas, right? And I think there's a nice image of this down here somewhere. Not for aspartate. Is there not? Like this, right? This is for phosphoric acid, right? This is what happens when you have a polyprotic, right? What you should note is that to go from here to here, I need one equivalent of hydroxide, right? It does not matter if my solution has 5 moles of phosphoric acid or 10 moles or whatever. It's still one equivalent of OH minus. It's just that the total amount of hydroxide ions in there in this specific case would be 10 moles, right? Does that sort of make sense? It's really weird. Yeah? Okay. So, are there any questions about any of that so far? Yeah.
Solution and now add 10 moles of base.
Mhm.
Just be adding 1 mole of
Yes, that is one equivalent, right? But just as equally, if I have 5 moles of acid in a solution and I add 5 moles of base, that is also an equivalent, right? It's just that I'm adding some amount of base or some amount of acid, right, to completely counteract the amount of base or acid already existed in the solution, right? It just has to basically take it from one pKa to the other, or one equivalence point to the other, right? So when we look at we look here, right, to go from this point here to this point here, I'm basically taking all of the H3PO4, the phosphoric acid, right, and turning it into the conjugate base form, right? In order to turn all of it from the acid to the base form, I need to add enough base to get rid of all the phosphoric acid, right? That is one equivalent, right? So that number changes depending on the molarity of the solution. Good? Yeah? Okay. Any other questions regarding that? Or do we need time to take notes before I move on? I feel like I'm moving kind of fast. Good? Okay. Trying to see if there's really another slide in this lecture that I want to go over. Did you guys do this question in class? The whole buffer thingy? Do you guys remember? How did we do on that? This buffer question, where is it? There's like a specific. Oh yeah, like this guy here. Do we feel comfortable with this idea? You want me to go over it? Yeah, okay, sure. So, I'm dying. Okay, so, what do we start with? We start with the fact that we're going over, we have an experiment that we're doing at pH 8, right? Where is pH 8 on this graph, right? pH 8 is right here, right? So if I draw a line across this, that's a horrible line, but you get the idea, right? I know that I have to be using either this black line or this red line, right? Does that make sense so far? Because we're looking for an ion of some sort that can buffer the pH that we're at. We cannot use this here because it's so far down, right? It's not even within the range, right? That makes sense so far, right? Okay. And even then, the question does say that you have ammonium phosphate anyways, right? So, but the key part of this question is that the experiment generates H+. Is that acidic or basic? It's acidic, right? Okay. If it's acidic, right, we're going to be decreasing in pH, right? But we want to make sure that the pH of our solution decreases as little as possible, right? If I add acid to this place right here, I'm going down this curve, right? That's a relatively steep drop, right? If I add H+ here, it's relatively flat, right? It goes down a little bit, right? But the pH change here is like what? I don't know, 7.86 to like 7, right? So it drops by a little bit, right? If I go here, though, it drops by like 8 all the way down to like 6.5, right? So that's a pretty big drop, and it would only drop further as I add more H+, right? So that's why we would probably pick this phosphate buffer and not the ammonium buffer, right? Good? Okay. What else do I want to cover here? Okay, yeah. They make this a lot more complicated than you need to know. This equation is a thing. The blood buffering system is the thing that keeps you alive, right? So you probably learned about this in 220, right? The blood buffering system is just this idea of CO2 plus water makes bicarbonate plus, right? This is an easy points on the exam, okay? My exam, the 2024 test, had 12 points that was just, can you do Le Chatelier's principle, right? So these should be easy points, right? You should be able to look at it and say, okay, it shifts this way, okay, it shifts this way, right? Because you will need to save time for the other stuff on the exam, right? But does anybody have questions about the blood buffering system? Yeah.
Not explaining this, but all the examples mostly multiple choice.
No. So I don't know if you've looked at the exams yet, but the 2024 exam was the test I took. I don't actually know if there was like any multiple choice on that. There might have been like a couple, but a lot of it is like, a lot of it is just like write out amino acid sequence, which one causes disulfide bonds, which one's hydrophobic. So that's the first page, and then the rest of it can ask a whole bunch of stuff. I don't remember a lot of multiple choice. There is some multiple choice, right? Like towards the back end, they might have some multiple choice questions. The Le Chatelier's principles one tend to be multiple choice, but it depends from year to year, right? Does anybody have any questions about? Yeah, I would memorize like this to this, but other than that, not really. The main thing I would keep in mind is just like CO2 plus H2O makes bicarb. That's that word too. And H plus. This is the main thing I'd memorize, right? This will most likely get you through the class, right? The one thing they love asking about is like, oh my gosh, you hold your breath and your CO2 goes up. What happens? You make more H plus and you get acidosis, right? They love those kinds of questions where they ask you like certain scenarios and then they'll go over that. They'll like, you have kidney failure, you have a lot of bicarb in your blood or something. How does that affect the the levels of things in your blood. Well, if you have a lot of bicarb, you'd shift to the left, right? You'd have a lot of CO2, right? So those are the kinds of things that they would test you on. What's up?
We have your test on like if your pH and making calculate that concentration of CO2 compared to like
Oh yeah, they could. Yeah, they definitely could. There's an equation for that. Well, that I'll go over in a second. But
well,
yes, you would use the Henderson-Hasselbalch equation. Yeah, you would have to use the Henderson-Hasselbalch.
outside of
Yeah, so this is where it's a bit, but yes, yeah. The whatever reason, Biochem doesn't care about the buffering range. But it is what it is. But any other questions regarding this? I feel like this is probably 220 stuff that we have like at least a little bit of knowledge about. So if I don't need to go over it too much, I won't. But we're all good here. Yeah, I would say just memorize this equation here, and you're probably set. Um, BS, I don't care. Again, so I mean, like, I guess for this is the equation that you're talking about, right? So this is what you'd have to use, right, for any kind of blood buffering question, right? And this is why I said that it's important to memorize this, right? Because it's just using CO2 and bicarb in the actual equation, right? But other than that, there's not really much else for this part here. Okay, this is actually a really good question. Why? Does anybody know why we're buffering at PK 6.1 and not 7.4?
body
Okay, it's a good start. Okay, yeah. Why does starting at a PK of 6.1 matter, though, for that? Okay, yeah. I mean, you have the right idea, right? I mean, because if we started a PK of 6.1, right, we can accommodate the body produces a lot more acid producing reactions than base producing reactions. So by starting at a pK of 6.1, when our pH is 7.4, if you remember that graph, right, of that buffering graph, right, um, let's just say this, right, if our pK is 6.1, right, and the pH that we're actually at in the body is 7.4, right, this line is like up here. So if I add a bunch of acid here, I'm just going to go down. That's really poorly drawn, but you get the idea, right? If I add acid here, I have all of this slope to go down, right? All of this slope with little pH change, right? That is why we have a buffer at 6.1, right? And not at 7.4. Because if we had a buffer at 7.4, right, you add acid and eventually it would start going down to this equivalence point where you get a pH of 2, which is what your stomach is at, right? And then you would die, right? Does that make sense? Yeah? Yes? Okay. I know I'm going a bit fast here, but hopefully that makes sense, right? Again, if you need more of explanations on these, the videos are available. Yeah, what's up? Yep.
said one the pH of body said
Yeah. So the pH of your body is 7.4 because that's the optimal temperature most of the enzymes, or sorry, optimal temperature, also pH that the enzymes operate at, right? Um, Other than that, there's other buffering systems. So there's the bicarb buffering system. If you study for the MCAT, there's the phosphate buffering system. There's a couple other buffering systems in there, but for the purpose of this class, you only care about the bicarb system. That's the main one. Yep.
there's a the body at 7.4 pH. Is there an other system that feasible?
It would be a combination of both, but bicarb is the main one that keeps it at that pH. I know that the pKa is not 7.4, right? But the bicarb buffer system is the main one that regulates the H plus concentrations in our solution, right?
Exactly.
No, they're not exactly the same. That's very important. Equilibrium does not necessarily mean 50-50, right? Equilibrium generally does not mean that actually, right? So that's what you should keep in mind. Yep.
So that doesn't like about like our pH of our body could be like 7.1, no 6.1 percent.
or like our pH around our values right now, right? Could be like in a range of 6.1 to 7.1. Like if you were like that.
Theoretically, yes, but you'll find out later that it's actually tightly regulated. It's actually like 7.2 to like 7.3 or like 7.4. It's a really tight range. And the reasoning for that has to do more with like the body's physiology than the actual buffering system itself. The body will not function anywhere outside those pH ranges because all the enzymes will denature, all of the organs will, like those cells will rupture, and you just get a whole bunch of other problems that will lead to death usually. So it needs to be tightly regulated in that range, and it's a combination of multiple things, right? Combination of the bicarb system, combination of the phosphate system, combination of your kidneys. Your kidneys help with that as well, right? Your kidneys help regulate that pH. And there's a bunch of other factors that contribute to that, but we mainly care about the bicarb system, right? For 440, they will only test you on the bicarb system when it comes to pH regulation in the body, right? Cool. Any other questions on this stuff here? Yeah.
I heard this mention that it's like a dynamic system. What is that?
It just means that it's so if the pH goes down, right, because of you hold your breath, right, um, the body will forcefully reconcile with that. So it'll force you to breathe, right? So if I hold my breath for a long time, eventually I will be forced to breathe, even if I try to stop myself from breathing, right? That's because we talk about it being dynamic because it's always moving, right? So if I slowly increase, where is it? So even if I increase the H plus concentration, it'll move left to counteract that, and I'll start breathing out, right? If for whatever reason my kidneys shut down and I have a lot of bicarb in my system, right? The buffering system will shift to the left, and I'll start breathing hard again, right? All they're saying by dynamic is that it's not static. It's not stuck in one place. It's always shifting, and Le Chatelier's principle always applies. Right. That's all they're saying. Good? Yep.
Professor, I think it's grad student, but she was saying that loss of bicarb
Mhm.
would actually no, 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 no. You loss of bicarb would cause a principal equilibrium to the acidic side, right?
Yeah. Yep.
So there would be out.
It'll be acidosis. Yep. Yeah, yeah. Yep. Yep. Good. Yeah, that's the kind of question they could ask on an exam. Right? That's a good way of thinking about it, right? They could easily ask that on an exam. Right? Okay. Any other questions regarding this? A big takeaway from this is if you see blood buffering and bicarb, this equation. Right? That's the equation you should be using. And yeah, so like you should be able to go through all of these, right? And tell me how this happens, right? So like hyperventilation, why does it cause alkalosis, right? So if I go back here, I'm too lazy to draw the double arrows. Someone tell me why hyperventilation causes alkalosis. This first one. Why is that the case?
Why CO2? Yeah.
Okay. Anybody else got an idea? Okay, so you're saying what? Breathe out a lot of CO2? So you're saying this goes down?
Yes.
This shifts this way? And you drop the H plus concentration, which means less H plus, more basic, alkalosis. Right? That makes sense? Cool, right? Okay, what about extreme prolonged exercise? What does that do? Why does that lead to this?
You have the God.
Yeah. Okay, that's one way of thinking about it. Yeah, that's also true. You produce lactic acid, and that goes out in the bloodstream, and that dissociates, right? What's another reason? Based off of this, what do cells produce?
CO2.
CO2. If I increase CO2, This goes this way, right? More acid, right? We know what apnea is. Yep. No? Okay. Apnea is like when your throat area closes and you can't breathe, right? So if you can't breathe, right, you'll build up CO2, right? This will increase, shift to the right. Acidosis, right? You should be able to reason through these in your head, right? And hopefully, when you see this on the exam, I don't know, when's your first test? I was gonna say, surely it can't be next week. Okay. Shouldn't have a heart attack. Okay, but yeah. Okay. That's quite soon, actually. Um, I wouldn't be surprised if this showed up for a couple of points, right? Yeah. Okay. Yeah, this will most likely show up, right? At least some form of this will show up, right? Because it's so easy to test on, right? And it's I wouldn't say basic physiology, but it's like physiology that like you can ask in 8 billion different forms, right? But it's just the same principle, right? So, yeah. Yeah, this is yeah. Well, this one has threw me off a little bit. I think the thing here is that if you lose HCl from the stomach, you're assuming the stomach ruptures and it goes into your blood vessels. But I'm actually not too sure how that works because that would suggest that it would increase the H plus, which would decrease the pH. But I'm actually not too sure how that one would work. I have to do research into that one. But if I had to guess, based off of just simply, I assume it's just decrease H plus alkalosis. That's the one that makes the most sense to me. They most likely will not ask you about that though. That's like very niche. They could theoretically just say loss of HCl from the stomach. How does that affect things? And for your guys' knowledge, I would just say that loss of HCl decrease H plus alkalosis. Right? But they need to connect this. I don't have access to your guys' like actual lectures, so I can't hear what they said. But they would need to explain this a bit more, right, for you to actually get an idea of what the answer would be. Okay? Good. Okay, does anybody have any questions about buffers in general? Buffers, um, any of the stuff that I've covered so far? No? Good. Like I said before, the videos are available. Those are much longer than this, and they're also a lot more in depth. So if you have questions based off of the videos, if you choose to watch them, feel free to come and ask me questions, and I can try and clarify stuff because those videos are kind of just made with me ranting. So yeah, um, I think we do have a little bit of time to go over the last one, lecture 3 at least. Lecture 3 is relatively short actually because there's not much I can tell you here other than just memorize amino acids. Um, but hello, welcome. Let's see here. Okay, so for lecture 3, before I even do any of this, I just want to say, I think I said this before, but just like the sooner you memorize the amino acids, the easier the class becomes. There's no shortcut to that. Memorize the amino acid name, the 1-letter code, the 3-letter code, the structure, and you should be able to, if you were given a string of 1-letter code amino acid things, you should be able to name every single amino acid. Tell me if it's acidic or basic. What's the pKa? At physiological pH, what is the charge of this peptide? Right? Obviously, you probably can't do that now, but by the time you're at the exam, if you're able to do that, you will absolutely destroy anything about amino acids on that test. Right? And also, that's what the MCAT does. The MCAT will just give you a long string of 1-letter codes, and they will just say, what's the charge of this peptide? What's the characteristic of this peptide? Hydrophobic, hydrophilic, negative, positive, what is it? Right? So, yeah. Um, let's see. Right, yeah. So here's the answer for that. Because if you wanted these, but we covered this though, right? Like hyperventilation, loss of CO2, right? We talked about. We didn't talk about this to be fair, but we talked about this, right? Increased CO2, right? Kind of talked about this, right? This is a bit of a weird one, right? But they literally just took it at face value, right? Same with apnea, right? We talked about closing the airway, right? Increased CO2, right? Um, I don't know what they were talking. Does anybody know what they were talking about for this stuff? Were they just yapping about protein models?
Yeah, they just let us.
Lo and behold, my microtubule guys. This okay, cool. I was gonna say because this stuff, if you're a biochem major, you have to take biochem 426. That's when I would care about like softwares and stuff like that, right? 426, it's the lab class, right? The lab class, you work a little bit with Primal and you start doing 3D structures of proteins in real time, right? It's cool. It's really cool. I love that class. Taught by Holt as well, who's unfortunately not teaching your class this quarter. I actually asked him. He's not teaching your class, but he was my professor and he's the goat. Um, but yeah, they just do like 5 slides of this, which is insane. Um, does anybody have overarching questions about amino acids that they want answered before I start jumping into all this stuff?
You go over the DHL naming system.
Yeah, that's a bit weird, and honestly, for like in-depth stuff, Scott is the best choice for that because he has a really good way of explaining it. Um, but all amino acids are L-amino acids, right? Um, I personally got by in 440 by just saying that L equals S and D equals R. That is not true for all amino acids. But I never had a case where I had to like do some weird shenanigans and switch it around. The only one that didn't apply to was cysteine. Cysteine is not S; it's actually R, even though it's an L amino acid, right? So that rule is not always true. Scott has a really good way of kind of deciphering whether it's D or L because that naming scheme is based off of glyceraldehyde. It's the same scheming as the sugars, right? So if you remember the sugar schemes, just keep that in mind. But other than that, they don't really test you on that. That's more of a 405, 406 thing where they start testing you on the D and L stuff. They will test you on buffers more likely than that. What's up?
It's like
Oh yeah. So there are uncommon amino acids. So there's selenium instead of cysteine, right? No, you don't need to know any of that nonsense, right? The 20 amino acids, the 20 common amino acids. That's all you need to know. If they give you a weird thing like selenium, they will tell you, right? So yeah, what's up?
We need to memorize which DR.
In 441, you will. In 441, you'll have to memorize which ones are. You'll have to memorize generally which ones are essential. But yeah, you don't have to memorize them now. But yeah. Um, other than that, yeah. Any other questions before I go into this? We have like 7 minutes, so I'll just briefly cover some of the stuff that's important. Yep. Yeah, so you guys have the quiz structure from our 442 class. We did not have in-person quizzes in 440 when I took it. They were online. But you guys have what we did in 442, where it sounds like you guys have a massive like problem set, and they pick one, right? Yeah, it's the exact same. If it is how I think it is, because in 442, the TAs would each make a quiz, and they would pick one of those quizzes. They'd make 6 quizzes. You'd get the quizzes a week beforehand. You do all of them on your own time, right? And then your TA would pick one of those quizzes for you to do during the quiz section, right? Yeah, that's fine. Yeah, I mean, as long as you're actually comprehending what you're writing is the thing, right? Because you don't want to just go blank. I will say that if you guys want me to go over those, I can definitely do that. I just need to get access to them, but Holt did just give me access to that Canvas page. So I can definitely try and go over them. But we shall see. I will do my best, though. But yeah, that's definitely annoying. 'Cause how many points are they? Are they 10 points each?
Something like that.
Okay, and it's what? Yeah, what's the other 90%? Yeah. Okay. Yeah. 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 Yes. That's. Yeah.
Yeah.
Yeah. Yeah. So I mean that tends to be how it goes. But yeah, hopefully your TAs just are nice about it because if you get a nice TA, it makes life a lot easier. Um, no, I'm not. I'm a tutor here. I don't TA that class. Yeah, I took that class last year. So I took the whole 44X series last year. But yeah, I just tutor here, and I just happen to have taken it. But yeah, I think I think the main things I want to get across here are like this whole PI thing. If you remember this from OChem, um, this is just the whole idea of a Zwitter ion, right? If you remember what a Zwitter ion is, don't. If you don't remember what it is, don't worry. If you do remember what it is, great. But basically, I will use this like lysine thing as an example, right? But the Zwitter ion is the point at which the charge of the amino acid or the peptide is zero, right? So the charge is zero, then there's obviously usually not going to be any interactions. But the easiest way to do this, if you're not comfortable or just don't remember how to do these, is to do a number line, right? And then to go through these. So the pKa of I'm just going to round these. I'm too lazy. This is going to be pKa1. This is 2, right? This is going to be 9, right? And then that's horrible. Oh my gosh. And then this is like 11, right? So we've drawn our number line, right? And then we can start going through this amino acid and seeing what is what at different pHs, right? So if we go all the way down to pH of 0, right, you're most likely going to be in the most protonated state possible, right? Nothing is going to be deprotonated, right? So the N-terminus, right, is basic. It's going to be positively charged. The lysine itself, the side chain, is an amine, also basic, positively charged. The OH group for the carboxylate is going to be protonated. It's neutral, right? So all the way down here is going to be +2, right? Does that make sense how I got to that? Yes? Yeah? Okay. Now, I don't even have to do anything else. I can just say, okay, +1, 0, -1. If you do that whole method, right, where if you go, okay, let's pick a pH between these two, 7, what is it? What is the charge of each individual group at that stage, right? You can figure out this is the correct answer, right? But the Zwitter ion stage is here, right? And the PI is just taking the two pKa's that are either side of this 0 charge, adding them and dividing it by 2, right? That is it. And lo and behold, it's the right 9.87, right? Now, the easy way to do this, without doing any of this nonsense, is just saying, if you have a basic amino acid, because if it's, usually you do single amino acids, right? If you do a whole chain of amino acids, this technique doesn't work. But if you have a single amino acid, just, if you have a basic amino acid, take the side chain pKa plus the N-terminus and divide it by 2. That'll pretty much always give you the PI of the basic amino acid, right? So like lysine and arginine, right? Take the side chain pKa and the N-terminus and divide it by 2, right? That'll give you the PI for that basic amino acid, right? Because that's what we're doing here, right? We're just saying this is the N-terminus, right? This is the N-terminus, and that is the side chain of the amino acid, right? We can do the same thing for aspartate, right? So a negatively charged amino acid, right? So that one's like 2, 4, and like 9, right? So this is a negatively charged amino acid, right? So we can do the same thing here, right? So if we go all the way down to pH of 0, it's going to be aspartate. The side chain is a carboxylate group, so it's going to be neutral. The N-terminus is positive, and the C-terminus is neutral, right? So all the way down at the very bottom, it's +1, right? And we do the same technique. 0, -1, -2. Notice, this is the C-terminus. This is the pKa of the side chain, right? So, for basic amino acids, all you gotta do is the side chain plus the N-terminus divided by 2. For the acidic amino acids, you do the side chain plus the C-terminus divided by 2, right? That's just a nice little trick that you can do without having to do any of the nonsense of doing the Celerion ion stuff, right? Cool?
I swear I did that on my work. I got
Oh, interesting. Okay, I guess was it a peptide chain or was it a single amino acid? Okay, well we can take a look at it. It is 7, by the way. Yeah, it is 7, by the way. Technically, it's supposed to be from 6 to 7, so you can leave if you want. I'm not gonna keep you here. But yeah, I don't think there's really anything else that I really ignore that. don't. I don't know why they showed you this. This is so intimidating for no reason. For these will become really intuitive as you do them, right? Like you'll go through it and you'll be like, oh, well, of course it's going to be positively charged, right? The things that have polar atoms are polar, believe it or not, right? So like serine is polar, threonine is polar, asparagine, glutamine, all these are polar, right? Yeah, don't no. Selenocysteine I don't care about. Yeah, proline, glycine, cysteine. We'll talk about these more when we need to. But no worries. And then all the hydrophobic ones just are either mostly hydrophobic with a benzene ring, or they just don't contain any other polar groups. That makes sense. Yeah, go for it. Yeah. Yeah.
It was
Yeah. So yeah, no worries.
It was tyrosine, but so it was this plus this because it's a
Yeah, because tyrosine. Yeah. Okay.
yeah.
Yeah. Okay. So
Which confuses me.
yeah. Okay. So this one we can do that method. We can see if it works, right? So let's go ahead and do space to do this. So for tyrosine, right? It would still be like what 2. Tyrosine is like 10. So yeah, and this is like 9, right?
So the side chain was outside of that range.
Yeah, I mean this is a bit of a weird one, but like we can go ahead and do it. So like yeah, if you're at 0, right? Visualizing it in your head is helpful, right? But obviously, do you have the amino acids memorized?
A little bit.
Okay, it's okay if you don't, right? No pressure. Yeah. But at pH of 0, the tyrosine would be protonated, so it's neutral. The amine group would be protonated, so it's positive, and the carboxylate is also protonated, so it's neutral, right? So plus 1 here, 0 here, -1, -2, right?
This is the carboxyl. This is the amine. That's why
Yeah, so that one's a bit weird, right? That technique works for the basic and the what's it called? The basic and the acidic acids, right? But yeah, that's a bit weird. Tyrosine is one of the ones that has 3 pKa's despite being not a base or an acid. So yeah.
Sorry, I wasn't super late.
No, you're okay.
But you do this every week.
Yeah. I'm hoping to hold it every week. It's like kind of new. I've never done this before, so it's like one of those great things. But yeah. There's a form as well if you want to fill it out. It's like just suggestions or like not suggestions, but like where is it? My ass is forgetting where it is. Yeah. There's a form here. It's for me to figure out if I'm actually helping you guys or not. Yeah. I wasn't sure because like I'm still a student, right? So like, but yeah, I'm a senior. Yeah. So, but yeah, feel free to fill out the form. There's questions in there. Yeah, you're fat. Fill out the form, broski. You showed up late and didn't fill it out. You filled it out. You're good. The form.
Oh.
Yeah.
Do must this turn.
No, you're all good. Yeah. Yeah. It just helps me figure out whether or not it's actually helpful if I do these or not. Because like I have to cover so much content in so little time, which is why I'm making the videos. The videos are way more. Was it good? Was it okay? That was my worry. Is like it's not helpful at all. It's just like that's crazy.
Some like tips and tricks.
Yeah. That's the main thing. Yeah. I was talking. In the last question in the what's it called, the form is like asking whether or not you guys want me to go over an exam on like how I would tackle the test. So that's also an option, but yeah, go for it. Yeah, take your time. Yeah.
Up and down.
Cool.
So you took that one out already?
Yeah, I did. Yeah. The MCAT's a tough exam, but it's made easier if you understand what you're doing. So, what I mean by that is like, how strong are your foundations in Gen Chem? How strong are your foundations in OChem? If your foundations are strong, then Biochem is really easy. And if your Biochem is really strong, then reading the passives are easy, right? So it all starts from the basics, right? If you build from the ground up, it's very hard to make mistakes, right? If you memorize everything and brute force it, you will lose points. You will lose points and you won't score well, right? So you must understand it, right? Especially because the MCAT is so broad.
Right.
The MCAT is so broad.
There's psych,
There's psych soc, there's reading, there's physics, there's OChem, there's Gen Chem, there's bio, there's biochem, there's physio, there's reproductive stuff. It's a lot of info. This cycle, yeah. But yeah, thank you, yeah. But yeah, that's what I would say. Let me